Thank you so much for that introduction, and thank you so much for inviting me to the epidemiology seminar series. It's an honor and a great pleasure, and it's a nice occasion after having worked mainly on COVID for so long to return to what has been a passion of mine for many years, namely to develop the whole country as a cohort. I will speak today about let me see if this moves. No, it doesn't. Why doesn't it? It did earlier on. There it is. I will speak about the power of cohorts and particularly about MOBA, the Norwegian Mother, Father and Child Cohort Study, because that is the cohort that I have been working on now for nearly 30 years. Also how that cohort relates to what I will call Norway's data ecosystem. I will give you some examples of users of the cohort. Those will mainly be from the COVID pandemic, so not about neurodevelopment this time, and a few future perspectives that are up for discussion. History is unfolding around us, and in just my or our lifetimes, we've seen unprecedented leaps in the scientific understanding of human biology, environment, and society. And these insights are enabled by new technologies, highly organized societies, and the entrepreneurial spirit of researchers and funders. Part of this is a revolution in the kinds of data available to epidemiologists who search for causes of disease and good health. So historically, and I'm sure many of you are aware of this, one of the most important examples of a high impact cohort study is that of Richard Doll and Bradford Hill, published in 1954. They linked data from a simple questionnaire about smoking among doctors to information about causes of death from the Registrar General in England. And this design was part of the reason why they eventually managed to convince authorities and the public that smoking in fact was a cause of death and disease. And this simple idea is what many of us have continued to work on since then. It turns out that it's very hard to establish population-based large cohorts and to run them for a long time over the life course of the participants, and not least over the sort of course of working life for many of those establishing them and running them. And still, cohorts are entrepreneurial. Most of them are not any routine infrastructures for research, which I have been hoping that they will eventually become in the future. So why is it easier to establish a cohort in a Scandinavian or Nordic country than in other countries? Well, that is what's been described in an article that I've more or less stolen the title from, called When an Entire Country is a Cohort, written by Lone Frank in Science in March 2000. And that's where Mats Melby, who was then an epidemiologist at the Danish equivalent to the Norwegian Institute of Public Health, where he describes how the registries and the twin study, which is a kind of cohort, constitutes a cohort using information from the whole population. So these cohorts, I think, have enormous potential for discoveries and for relevant use in society. And the achievements that have been made so far are impressive, but I still think the most important discoveries lie ahead. And I also think that MOBA, the Norwegian Mother, Father and Child Cohort Study, is a most promising source for epidemiological analysis in the future. So to the details of how MOBA and the Norway's data ecosystem looks. MOBA is population-based. It's a large sample with a long follow-up, and we link it regularly to registries that are mandatory and population-wide, and we have biobanks as part of MOBA. The setting is that the whole country is a cohort, that we have a national ID number that all inhabitants in the country have, whether they're born here or have immigrated. And that we have developed scientific epidemiological competence and capacity, not least because many people are interested in these kinds of sources and move to Norway to be able to work more closely on them. So key features of MOBA are that it's a family cohort where we recruited first mothers, then fathers, and children. But it turns out that many more are related with each other. For example, there are about 1,800 twins among the children that were born into the cohort. Many of the mothers turn out to be sisters, and some of the fathers turn out to be brothers. And also there are other family relations that we have now identified in the whole MOBA cohort, which means that we can study both the genes and environment, and we can use both genetic and environmental information in analysis. Recruitment of pregnant women happened from 1999 to 2009. I will return to the details of that in a minute or in a moment. Altogether, 95,000 mothers, 75,000 fathers, and nearly 115,000 children have been recruited, and most of them are still following up. Very few have left the cohort, although after the pandemic, there have been a few more than usual, but extremely few compared with most other large cohorts internationally. They followed up with questionnaires, biomaterials, and registry linkages. And that means that we have lots of exposures and many outcomes in the cohort, and not all of that has been used or exploited sufficiently yet. And it's accumulating all the time because new sub-studies, they return data to the main cohort, which means that it's increasing in value, scientific value, and otherwise continuously. About 250,000 samples have been genotyped so far, and GWAS, genome-wide association studies, is available in 70,000 trios, mother, father, and child trios, and an epigenome-wide association analysis is available in 15,000. So here's a bit more detail on the recruitment and the follow-up. You see to the right here that we did recruit over 10 years. That wasn't because we had planned for it. It was because of logistical challenges, political challenges, and funding, mainly funding, but also these other factors. However, it turns out that that is an asset for the cohort because we can now study what happened with the first part of the cohort and compare with the later part of the cohort. And that means that we have been using it to look at secular trends, natural experiments, for example, the introduction of pneumococcal vaccination for children at the age of 2 years in the midst of recruitment, and many other things that have changed over the 10 years we recruited. Also, it means that we have representatives from many different age groups, which has been an asset during the COVID-19 pandemic, for example. You can also see here at the sort of middle of the timeline, in 2015, we were eventually able to do a second round of data collection by questionnaire from fathers. And also we have been following up fathers in some sub-studies. Now the yellow column here is the COVID-19 pandemic. And we see that that happened in a very sort of limited time period And we decided early on that we would use this cohort and other cohorts in studying and following up on the effects of the pandemic and the interventions or the measures that were used, and not least vaccination. So I'll return to that. The general form of the ecosystem or health data system can be seen in the figure to the right here, because all inhabitants are part of registries, and then subgroups, larger and smaller, are parts of cohorts. And because we have many other large population-based long-term cohorts in Norway, such as the Hunt study, the Tromsø health study, etc., it means that more than 10% of the population is being followed up in in-depth cohort studies with biobanks and with linkage through the personal identification number. And to the left here, you can see that there are many different kinds of registries, and I won't go into the details of all of them, but we do, for example, have information from specialist care, from hospitals, from primary care, on all prescriptions, on vaccinations, on infectious diseases, and on cancer, cardiovascular diseases, induced abortion, and also nursing and care. And we have newborn screening, which we haven't been using to a large extent yet, but we're hoping to link it to the cohort data as well. So this is just a reminder that we have a sub-project called MOBA Genetics, where we have all the genotype data, but also related data such as methylation, telomere length, metabolomics, sequencing data to some extent, but not as much as we wish, and mitochondrial DNA analysis. This is a picture of what is called the Emergency Preparedness Registry for COVID-19. Because a major limitation in our use of the MOBA cohort and the registries is that it can take a very long time before we get access to data through linkage. And although that has been an issue for more than 20 years, it hasn't really improved. But during the pandemic, we had this special law that allowed us to link all kinds of data. And you can see all the different kinds of data that were linked. Actually, that isn't complete to the left here. For example, we had daily updates on positive COVID-19 cases from the Infectious Disease Registry. We had daily updates on vaccination of COVID-19, influenza, and pneumococcal vaccines. And we had hospitalization data with intensive care information as daily updates. And of course, we had daily updates, or not that often, on the population registry, so that we would always know how many people that were included in the whole population at the time. So there were also many other kinds of data than health data. There were data on occupation and workplace, on residents of old age and nursing homes, and on age, gender, municipality of residence, family relations, and country of birth. And country of birth turned out to be a very important variable during the pandemic. So to the right here, we see a few of the kinds of outcomes that have been studied and that we are still following up on. For example, infections in schools, complications after COVID-19, and the effects of vaccination and adverse events after vaccination. So we have also been discussing how we can use this Norwegian health data system in clinical trials. And although I won't go into that, I think that is a very interesting way of using cohorts in the future together with registry data. We can, for example, use the registry data and the cohorts to identify and quantify groups of special interest, for example, healthcare personnel, severely immunocompromised persons, nursing home residents, 4 different kinds of clinical studies that can be embedded in the cohorts and the registries and followed up over a much longer period than the more intensive clinical trials usually go on. So this is just an example to illustrate those potential users that haven't been exploited much to date. And this is an example with a figure to the right showing how we can use the registry data in follow-up of such clinical trials, much the same way as we've been using them in following up on vaccine safety and effectiveness studies during the pandemic and after the pandemic. This is just an example that I will return to with the number of emergency hospitalizations for myo and pericarditis, where there was a change in vaccine recommendations, which turned out to reduce those numbers very efficiently, almost overnight, for everyone under the age of 40. Now, sorry, I've said this already. In the future, we think that nested studies within population-based cohorts and registries of vaccine safety and effectiveness can be used in a much larger, to a much larger extent, and particularly if the Nordic countries can collaborate with each other, because that means that the total population will be about 27 million people. And that means that one could do such studies quite efficiently. So I've added nested randomized controlled trials to the general depiction of the health data system. Now to some examples. We do have examples of study designs using MOBA, and there are plenty of them. And I've just very short reviews of some of them that I've been directly or indirectly involved in myself. For example, we did a case cohort study, which was published in JAMA in 2013 on intake of folate supplements in early pregnancy. And we could show that it was associated with a 50% reduction in the occurrence of autism, but intake of fish oil supplements was not associated with autism, which was reassuring. But this hasn't really been replicated. So although I think it's still an interesting finding, we don't really know whether it is true or not, and whether it can lead to prevention of autism or not. So another study, they would use discordant sibling design and looked at paracetamol, which was used in one pregnancy, but not in another with the same mother. And we could show that paracetamol was associated with increased risk of neurodevelopmental disorders in children. And then a study that I've already mentioned, where there was a natural experiment, or the so-called natural experiment, to the extent that introducing a new vaccine is natural, where pneumococcal vaccine was introduced in 2006 in the middle of the MOBA inclusion period, and we could see a decline in early childhood respiratory tract infections. In fact, one could also later on observe a decline in pneumococcal infections among grandparents. And then to examples from the pandemic. We have been using cohorts during the pandemic to look at all these different kinds of outcomes that you can see here in this circle. I won't have examples on all of them, but there are many publications and many more will come. So we also supplemented the MOBA study with 3 other cohorts, because we needed coverage in the population that was broader than what MOBA could provide. So we had the Norwegian influenza study with 9,000 participants, mothers and children, where all children were 12 years, and they were recruited and followed up since the swine flu pandemic in 2009-2010. We also had a senior cohort with 5,000 participants aged 65 to 80 years, and a young adult cohort. This is just to show you how we could use cohorts together with the registries during the pandemic. Now, 1 year after infection, we found that 13 of 22 symptoms that people reported were associated with SARS-CoV-2 infection based on relative risks between infected and uninfected subjects. So this is our take on looking at what many will call long COVID, but we are questioning that term because our results suggest that there are clusters of symptoms after COVID-19 due to different mechanisms, and we are asking whether it is meaningful to describe long COVID as one syndrome. This is just one of the first publications on long COVID that came out a year ago. Soon after, vaccination had started. On the 11th of March 2021, we heard from the Danish public health authorities that they had a case of a woman who died soon after having been vaccinated with the COVID vaccine from AstraZeneca. That report worried us because it was such a very special syndrome that this woman had had. And we decided within hours that we would pause the use of that vaccine. At that time, we had just started vaccinating healthcare personnel, and most of them were women. We had been vaccinating them only for about 2 weeks, but we were fearing adverse events. And this was the first report that really made us worried. A few days after, or immediately after the report from Denmark, when they also had paused their use of the same vaccine, we set up Nordic collaboration to look at particularly adverse events from vaccines. That has turned out to be a very fruitful collaboration. There are similar types of registries that are updated regularly in all the Nordic countries, and there is single validation in the Nordic countries of the reports that, at least in Norway, anyone can turn in a report, the population, healthcare personnel, and others. So we can, by using the registries, see if the signals can be validated or not. And combined Nordic analysis means that we can look at very rare events. So a common protocol for analysis was set up. And both Denmark, Norway, and Iceland suspended the use of the AstraZeneca COVID vaccine over reports of this blood clot syndrome called VITT. And we followed up immediately in the first cycle only with Norwegian and Danish data after Denmark had announced that it was to temporarily suspend the use of the vaccine, and that it was important for us to emphasize that we had not opted out of the AstraZeneca vaccine, but we were putting it on hold, and that we would try to find out scientifically whether there was a reason to keep it on hold or take it out of the program entirely. So a few days later, after the event on 11th March 2021, the Danish-Norwegian collaboration was in place. And a few days after that again, combined results were finalized, and a paper was submitted already less than a month after the first event. And on the 5th of May, less than 2 months later, a paper was published. So that resulted in the Norwegian Institute of Public Health recommending that the AstraZeneca COVID vaccine should be dropped altogether from our vaccination program. That was an incredibly difficult decision to make because we really needed those vaccines at the time, but we had done studies showing that the risk for those who received the vaccine, who were young and healthy, was higher from this adverse event than it was at that time in Norway from COVID-19 itself. So this is a picture to the right here of the first publication, and we followed up then later on by including other countries as well. I haven't taken that with me, but this has been validated with data also from Sweden and Finland, and that means that we now feel even more confident that it was the right decision to make in our countries at least. Now, that collaboration has been followed up in looking at mRNA vaccines and myocarditis. And there were news reports from Israel, not scientific reports, and some spontaneous reports from doctors. There were very few events observed in registry data, and there was an urgent need for studies. Vaccinating thousands per day, as we did at the time, also younger age groups, meant that we were alerted to adverse events that could be prevented. Also, this resulted in a huge study with data from almost all the Nordic countries together, and that was important in being able to prevent further risks from myocarditis in young men in particular. The summary results from that combined Nordic study was that the vaccine type Moderna provided a higher risk than the Pfizer vaccine, that the second dose provided a higher risk than the first, and that younger people had a higher risk than older, and males higher than females. And also, but more uncertain, that using both vaccines provided a slightly higher risk than using only one vaccine in the two doses that were given at that time. The results were very consistent across Nordic countries. And again, that meant that we could provide advice very rapidly after the first events happened that were solidly scientifically rooted. Now, these are the examples that I've brought with me. They could have been so many more, and they are clearly influenced by the fact that COVID has been one of the most important tasks for me for the last 3 years. But we have been using the cohorts and the registries for many, many different kinds of studies. And one of the most interesting things that I'm doing myself at the moment is to use the MOBA cohort and the registries, not only the health registries, but registries on educational attainment and outcome on schools and school results, to look at whether the gender differences in education can be caused by the differences or the main differences and the distributions in development when it comes to skills that are important for results in school, particularly at the age of 15 and 16, which is a very important stage where many choices are made. So we are looking at whether this can explain the gender differences. And particularly, we are looking at the influence or the importance of differences in timing of puberty between boys and girls. We don't have all the results yet, but that will come later on. It would have been impossible to do these studies unless we had had the kinds of data that we do in the Nordic countries. And we are working with Denmark on these issues. Now to a few future perspectives. George Davies-Smith at Bristol University has compared the Nordic epidemiological birth cohorts in particular, which MOBA is, and the systems that they are part of with these registries with CERN's Large Hadron Collider, which is a physics infrastructure, which means that one can do extremely ambitious physics experiments. It's an infrastructure that all the different countries that are involved can use together. It seems that it's been much more difficult to get that kind of investment in the cohorts than it has been in the Hadron Collider. So what I wish for is that our governments, our politicians and our public and the media will understand how valuable cohorts and registries are, and that we should invest in them to make them safe, to make them secure for individuals, but also useful for individuals as well as for science and for society. It's been possible to invest in science, however, when it comes to the use of those registries and the cohorts, although they themselves have been struggling very hard to get funded. But that has been possible because of international scientific collaboration and interest in particular usages, particular aims. These are platforms that enable the pursuit of many different research questions. And as I said initially, they do become more and more valuable as time passes. Now, how can we unlock the potential of MOBA? For us, there are currently three main challenges. We need large-scale genetic analysis with sequencing data. We need new infrastructures for data storage. We need a modern regulatory framework that handles the complexities. And particularly when it comes to the regulatory framework, that is not just an national issue, it's also an international issue. And my personal opinion is that in order to get that kind of regulatory framework, we also have to address the issue of how value, and I mean value in the sense of economic value, that comes out of or can come out of those kinds of resources can be returned to the populations, the societies, and sometimes even to the individuals that contribute to these kinds of data. Because unless that happens, it's going to be very hard to argue that it should be shared to a much larger extent than we are sharing it at the moment. So I think there is enormous potential for science, for individuals and for society and for better lives by developing such infrastructures further and using the ones we have and recognizing how important they can be, that they can be cost efficient, that they can lead to commercial products, to industries, and be used in ways that are not limited to epidemiological research and methods that we are using at the moment. We have seen how important these kinds of data are for security, for emergency preparedness and response. And I think that it could be a good way of illustrating how important it is, not only during crisis, but also during regular times. We are certain that these kinds of data, these kinds of infrastructures are important for public health, for health promotion, prevention, and protection, because we need much firmer science in order to work in public health and a much closer relationship between the science and between the policies and interventions that are chosen and the way they are followed up. So by those perspectives, I'm ending my talk, and I hope that there will be many questions. And if you don't raise the questions here, you are welcome to send me an email with questions or requests. And if you're interested in using MOBA and the other cohorts in Norway and the registries, you can also send me an email. I cannot promise you, though, that it will be a short time before you get access to data, but they are in principle available for all researchers all over the world if they collaborate with someone in Norway. Thank you very much.